Question number 92. Five kingdom system of classification suggested by R.H. Whittaker is not based on. स्पेशली पूछा गया नोट बेस्ड ऑन. अगर इस क्वेश्चन के आंसर की अगर हम सबसे पहले बात करें तो इस क्वेश्चन का आंसर होगा फर्स्ट. अब थोड़ा इस क्वेश्चन को समझने की ट्राई करते हैं. देखिए ऑप्शन नंबर 2, 3 एंड 4 ये तीनों विटेकर के क्लासिफिकेशन के क्राइटेरियाज हैं. अगर हम आपसे बात करें ऑप्शन नंबर 2 की, मोड ऑफ रिप्रोडक्शन, इसको विटेकर ने यूज किया अपने क्लासिफिकेशन के लिए. मोड ऑफ रिप्रोडक्शन के बेस पर विटेकर ने प्रोकैरियोट को एंड यूकैरियोट को डिफरेंशिएट किया, क्योंकि प्रोकैरियोट में हम सब जानते हैं कि सेक्सुअल रिप्रोडक्शन नहीं होता है और यूकैरियोट्स में होता है. तो मोड ऑफ रिप्रोडक्शन के बेस पर विटेकर ने मोनेरा को या कह दीजिए प्रोकैरियोट को यूकैरियोट से डिफरेंशिएट किया. इसलिए ये एक क्राइटेरिया है विटेकर के क्लासिफिकेशन का. अब हम आते हैं मोड ऑफ न्यूट्रिशन. मोड ऑफ न्यूट्रिशन के बेस पर विटेकर ने 3 मेन किंगडम को डिफरेंशिएट किया, माइकोटा, प्लांटी एंड एनिमेलिया. इसलिए ये भी विटेकर के क्लासिफिकेशन का एक इम्पोर्टेंट क्राइटेरिया है. अब हम बात करें कॉम्प्लेक्सिटी ऑफ बॉडी ऑर्गेनाइजेशन. देखिए इसका ऑप्शन का जो मीनिंग है वो है कि ऑर्गेनिज्म यूनिसेलुलर है या मल्टी सेलुलर. इस क्राइटेरिया का भी विटेकर ने यूज किया अपने क्लासिफिकेशन में. विटेकर ने ऐसे ऑर्गेनिज्म जो यूनिसेलुलर हैं उनको प्रोटिस्टा में रखा और ऐसे ऑर्गेनिज्म जो मल्टी सेलुलर हैं उनको माइकोटा, प्लांटी, एनिमेलिया में रखा. यानी कि ऑर्गेनिज्म यूनिसेलुलर है या मल्टी सेलुलर है, इसका विटेकर ने यूज किया अपने क्लासिफिकेशन में, या कह दीजिए बॉडी ऑर्गेनाइजेशन का यूज किया विटेकर ने अपने क्लासिफिकेशन में. तो इसलिए तीनों विटेकर के क्लासिफिकेशन में यूज में लिए गए. अब हम आते हैं फर्स्ट ऑप्शन की. देखिए इसमें लिखा हुआ है यहाँ पर कि प्रेजेंस और एब्सेंस ऑफ वेल डिफाइंड न्यूक्लियस. देखिए विटेकर ने अपने इस क्लासिफिकेशन में, या कह दीजिए विटेकर ने मोनेरा, प्रोटिस्टा, माइकोटा, प्लांटी, एनिमेलिया को डिफरेंशिएट करने के लिए डायरेक्टली न्यूक्लियस का यूज नहीं किया था. इन्होंने यूज किया था कंप्लीट सेल स्ट्रक्चर का. सेल स्ट्रक्चर को बेस बनाया था, नॉट ओनली न्यूक्लियस. इसलिए ये ऑप्शन इसमें क्वेश्चन के बेस पर सही होगा. यानी कि ये तीनों विटेकर के क्लासिफिकेशन के क्राइटेरिया हैं और ये डायरेक्टली क्राइटेरिया नहीं है, क्योंकि विटेकर ने सेल स्ट्रक्चर को बेस बनाया था, नॉट ओनली न्यूक्लियस को. थैंक यू वेरी मच.
We are going to discuss the question number 106, and the question is that during which phase of cell cycle the amount of DNA in a cell remains at 4C level if the initial amount is denoted as 2C. To discuss the question and to explain the different options given, we should go a brief outing of the cell cycle that is given here. We know that cell cycle during S phase the amount of DNA becomes double. If the initial amount which is given as 2C, then it will become 4C, and that will remain unchanged during G2 and M phase. At the end of N phase, the cell will divide and the amount will become half. So the correct option will go 4, that is G2 and M phase the amount will remain 4C and the amount will become half in G1 phase. So the correct option out of that, option number 1 G0 and G1 is wrong. Option number 2 G1 and S is also wrong. Option 3rd only G2 is also wrong, and the correct answer is G2 and M phase. So the answer will go 4th.
Question number 108. Dr. F.W. Wendt noted that if coleoptile tips were removed and placed on agar for 1 hour, the agar would produce a bending when placed on one side of the freshly cut coleoptile stems. Of what significance is this experiment? Here we are watching that certain apices of the Avena coleoptile are placed on the agar block for the 1 hour. Afterwards, we remove these coleoptiles and the agar blocks are cut it down into the small pieces. Now, these small pieces, whenever placed onto the stump of the coleoptile on one side, we are watching that there is a curvature. There is a curvature into the coleoptile. It means after making such a kind of a placement of the tips or apices of the coleoptile on agar block, we are capable to make a quantitative analysis of the oxygen. So if we are considering oxygen concentration on the one axis and curvature or degree of curvature on the another axis, we are watching that as per the concentration of oxygen, there is a very good curvature into the coleoptile. So as per the concept, it is made possible in the option number 1, it is given that it is made possible the isolation and exact identification of oxygen. It is not up to the mark, so we should shift to the another option, and this one is the, it is the basis for the quantitative determination of the small amount of the growth promoting substances. It means such kind of a agar block mediated experiment that was carried out by the AIPMT was one of the fabulous job specifically for the quantitative analysis of the oxygen. So as per the question, agar block mediated experiment was actually meant for the such kind of a quantitative analysis of the growth substances.
121. Which one of the following is wrongly matched? Here the 4 options are given, and out of these 4 options we have to understand that which one is wrong and which one are correct. Out of these 4 options that are given, 1, option 2, option 3 and option 4, we have to discuss that out of these 4, 1 is wrong and 3 are correct. First of all, we will discuss the option number 2, the translation, that is translation, using information in mRNA to make protein. That is correct, because during translation the informations which are received by RNA from the DNA, they are converted to the protein, so option 2 is correct. Regarding option number 3, that is repressor protein, binds to operator to stop enzyme synthesis. This option is also correct, because during that repressor protein binds to operator to stop the enzyme synthesis. Now we are going to option number 4, that is operon. That also include that structure genes, operator and promoter. So here out of these 4 options, option number 2, option number 3 and option number 4, they are all correct. Out of these 4, the answer will be that one, that is the wrong statement. Now we are coming to the first, that is transcription. Actually during transcription, entire raw material or a foundation is laid for the synthesis of protein. But this foundation or this process is only in coded language, it is not in the written informations. So whatever is given here, that in transcription, writing information from DNA to tRNA, informations are not given in a concrete or decoded language, it is in coded language, so this option is wrong. So answer will go 1. Is it clear? So in question number 121, which one of the following is wrongly made? That option number 1, transcription, writing informations from DNA to tRNA, it is wrongly made, and other 3 options like translation, that is using information in mRNA to make protein, it is correct. Third is repressor protein binds to operator to stop enzyme synthesis. This is also correct. And fourth one is operon, that includes structure gene, operator and promoter. It is also correct. So all three options, second, third and fourth, are correctly matched. Only wrongly matched option is option number 1, transcription, writing information from DNA to tRNA. So answer will be 1. Okay.